from creating cancer or that early detection of cancer to a new body. I mean, you know that cyborgs are quite old for makers. We are entering a new era, an era of engineered tissues, and the era is told by Nina Maria Tandon.
Good afternoon, everybody. I'd like to introduce you to Alana. When she was 11 years old, she was playing basketball, and she fell and shattered her kneecap. This was really dangerous. She lost a lot of blood. Surgeons were able to restore her knee, actually. They gave her a new patella from a donor. They refashioned ligaments out of her own ligaments, and they held everything together with biopolymers and metal screws. And she's really grateful about this. But she'll also be the first to tell you that this is not how people's bodies are going to be put back together in the future. We can imagine a future where instead of taking parts from other people and taking parts from technology, that we can use our own cells to grow new body parts for ourselves. How did we get to this idea? Up until very recently, we thought of the body as kind of an intact unit. This is Leonardo's Vitruvian Man. And basically, when something was wrong with the body, we kind of left it alone and hoped that it would heal itself. But somewhere about 100 years back, we started thinking, well, if a part of your body is broken, let's take a donor part, like if you need a new heart, get one from another person. Or if you need a new knee, let's make one out of titanium and ceramic and put it in your knee. But now, going forward, we're actually at a really crucial point in history where we can think about growing new parts for the human body using our own cells. And this is what I like to call Body 3.0, an era where we're going to use the power of our own cells to regenerate our bodies. And central to this idea is the idea that our cells are like Legos. Our cells are like living building blocks. And in fact, if you think about your own baby picture, I mean, we all looked like this at one point, that we started out as one cell, and that one cell divided and differentiated and became every kind of cell in your body. Brain, heart, liver, skin, lung, all of those came out of that one cell. So as biotechnologists, we ask the question, how did that happen? And secondly, how do we copy that in order to be able to grow new body parts in the lab? Okay, so this is the idea of tissue engineering, that we take cells, we put them on biomaterials, and we grow them in bioreactors, which are like fancy fish tanks, you know, telling the cells what they need to grow and giving them the nutrients they need, so that eventually on the other side, after 3 weeks or however long it takes, we can grow a new heart, grow a new bone, grow a new liver, and so on. So I wanted to give you a couple examples of what we're doing right now with that and what we could do with that in the future. So this idea of body 1.0, 2.0, and 3.0 is applied to the heart was what I did during my PhD research. And this is a picture of the bioreactor that we developed in order to do that. If you only knew 2 things about the heart, right, what would you need to know? One is that the heart beats rhythmically, tirelessly, faithfully, billions of beats in a lifetime, never pausing to rest. So not surprisingly, that makes heart cells pretty greedy. So they need to beat. So we need to apply electrical signals in the lab to get them to beat, but we also need to give them lots of food as we grow them. And so this is a bioreactor that gives those heart cells exactly what they need: electrodes to give them pacing and lots of tubes to be able to flow food through them. And this is a picture of an electrical stimulator that we used in our first research in this. And it cost about $3,000. But I wasn't satisfied with this, so on the side I started developing new stimulator circuits. This was using a computer, and now we're using circuits like this, using Arduinos. So the cost of doing this research is coming down as these technologies are coming online, and this is where I think makers can intersect with biotechnology. This is an example of a beating heart tissue that I engineered during my PhD, and as you can see, it's beating. It's beating in time with that stimulator circuit that we built. If you turn the dial for the frequency, the cells speed up. And if you turn the speed down, the cells slow down. This was an incredible idea for me, this idea that we could build a circuit that could speak to living things. What I'm involved with right now is actually a startup to apply the same principle towards bone. So bone 1.0, 2.0, 3.0, we're moving to a point where we can imagine putting our bodies back together using body parts that we grow out of our own cells. We call it EpiBone. We just started, and this is a really fun journey. This is how we grow bone. So we take cells, we plate them in flasks, and we grow them. These cells come from adipose tissue, so from fat tissue, and we take the stem cells out of them. So we grow them in the incubator for a couple weeks, and then we put the cells on a biomaterial scaffold in a bioreactor. We make the shape of the bone based on the patient's CT scan, so we can get the perfect shape using software to map out their skull, and we also use cells, so we get a perfect match in terms of the implant itself. So we put these in the bioreactor, and after 3 weeks in culture, the bone matures and turns into an equivalent to the living bone that you have in your bodies right now. And so we're hoping that in the future we'll be able to implant bones like this into the body. What else can we do with tissue engineering? Well, if you think about the drug screening process, for example, it costs over $1 billion for a drug to get from drug formulation through lab testing, animal testing, clinical trials, and finally getting to the market, right? It takes over 10 years. And a lot of times when drugs hit the market, they act in an unpredictable way and actually hurt people. And the later the drug fails, the worse the consequences. And it all boils down to 2 issues. 1, humans are not rats. And 2, despite the incredible similarities between all of us, those tiny differences in our DNA actually have huge impacts with how we metabolize drugs and how those drugs affect us. So what if we could use tissue engineering to make better models of the human body that act more like humans than rats, but also reflect our diversity? In order to be able to actually use these for replacing clinical trials, we're going to need to be able to grow these tissues in a more scalable way. And this is a schematic of a bioreactor we're developing in our lab to do just that. And remember Alana? She's actually a master's student who's working on this project. So she's very opinionated, but she has a right to her opinions. This is a schematic of how we actually run these bioreactors, how we're beginning to scale them up, and we love using as open source hardware whenever we can. In this case, we're using Arduinos to be able to deliver that stimulation to the cells, to be able to deliver everything that the cells need, but also to be able to read out from the cells what is their pH, what is their glucose, and so on, and coordinate all of that. And one of my students, Adrian, actually programmed an Android phone to be able to control the bioreactor. So we love open source hardware, and we think that, that this is a key technology innovation that can help us speed up our innovation. This is what the system looks like in practice. And just speaking of the cost of technology, when we make all of those little pieces of beating heart tissue, for example, we want to be able to do things like, well, do a mini EKG and see how the heart's doing, right? And there are systems to be able to do this. They cost about $100,000. This is on the left, a picture of a microelectrode array, MEA for short. And on the right, we have to use microscopes to be able to image. If you put that together, that costs over $100,000, and that's a lot of money. But what we're doing right now is we're taking webcams and hacking them apart and putting the cells on those to replace a microscope. And we're making our own circuits from scratch. And so we really believe that we can start to use, as technology on the consumer market gets better and better, to be able to use some of that technology in the lab to be able to bring down the costs and increase the scalability of what we're doing. Here's an example of an inkjet printer that Wei Wang hacked to be able to print cells, okay? And in Karen Berg's lab at Clemson University, they're actually using this technology to print breast cancer cells. So you can imagine using tissue engineering techniques in combination with some of these hacked apart technologies that people are developing in hacker spaces to be able to combine those and be able to test how a drug might work on your cancer, as opposed to just taking a guess, right? So we're really at a point now where we can imagine some amazing things. And so hopefully going forward, we can imagine this world where instead of clinical trials being tested on animals and people, that we can use technology to grow better models in the lab to be able to reduce those costs. Okay, so like a clinical trial on a chip. But what else can we do? Okay, because this is a maker audience, right? So I can't resist the idea of showing some really crazy stuff. So sure, body parts, right? But that's just the beginning. Okay, if you think, for example, that electric eels give more current than what we can do with batteries these days. This is an example in Japan where they're lighting Christmas trees from electric eels, right? So just thinking about tissue engineering, right? If we combine some of the advances with what we're doing, with what's happening in synthetic biology, maybe we can imagine making batteries out of cells. Why not? Maybe we can imagine making meat from cells. Instead of growing grass to feed our cows to make meat, why don't we skip all that and grow meat from scratch? And then, of course, the work of Oren Katz growing a victimless leather jacket. There's so many products on the market that use animals as the first step. Why don't we start growing leather from scratch using the power of cells? But how are we going to get there, okay? And I really think that, this is such a great audience because you guys know better than anyone else how to take apart what already exists, remake it into something else that you never imagined, right? But biotechnology is often a little bit presents more of a hurdle for us to be able to get into it. So I just wanted to offer that there are new spaces popping up all over the world. This place is in Brooklyn, GenSpace, and Nurit is going to talk in a couple minutes a little bit more about GenSpace in particular. But this is a biohacking space. It's the United States' first biohacking space. And what is that? It's a space that has all of the scientific tools available to be able to do biotechnology, but without confining us to having to use those technologies to answer scientific questions that academia would be interested in. So basically, it's like a hacker space for people interested in exploring biology. And this is a picture of me with Aaron, Steve, and Eliza, who are architecture students interested in growing sculptures out of skin that we transfected to glow green. So they call it their living tattoo project. And it'll be on display in Orléans later this year. What else can we do to increase this generation of biomakers? Okay, this is these are two high school girls that I was teaching how to extract DNA from out of strawberries, okay? And so, I just wanted to end with a couple thoughts. I hope I've convinced you, one, that biology is technology, and two, that hackers and makers make great biotechnologists. But thirdly, as we consider this question of how do we remake the world, if the first industrial revolution was about machines, and the second industrial revolution was about information, as we think about how to remake the world, let's make the third industrial revolution about life. Thank you so much.
Great, thank you.
Thank you.